Hi, my name is Zoe, and today we will be talking about how SARS-CoV-2 replicates its genome for packaging into new virus particles. Once inside cells, SARS-CoV-2 must replicate its genome to be packaged into new virus particles. Its genome is made up of single-stranded positive sense RNA. Positive sense means that the RNA is messenger RNA, ready to be directly read and converted into proteins. Conversely, negative sense RNA is complementary in its base pairs to positive sense RNA and therefore cannot be used as a code for making proteins. It instead serves as a template for making more positive sense RNA strands. SARS-CoV-2 replicates in human host cells, which are eukaryotic cells. Human cells, like all other living organism cells, contain their genetic material in the form of double-stranded DNA. Now, we will compare and contrast the process by which human DNA is converted to proteins with how the virus's RNA genome is converted to proteins. The conversion of DNA into proteins is known as the central dogma of biology, and in humans, it consists of two steps. The first one is transcription, in which the genetic material of a cell is used as a template to make RNA that carries the genetic instructions from the nucleus to the ribosomes. At the ribosome, the genetic message carried by the RNA is translated into proteins. This is the second step of the central dogma, and it is known as translation. SARS-CoV-2 undergoes a similar process to make proteins out of its genetic information. As opposed to DNA in humans, the virus stores its genetic information in an RNA genome. When the virus transcribes its genome, it makes a negative sense strand of RNA. Then, it makes more positive sense strands of RNA that are complementary. Some will be packaged into new virus particles, while some will be sent to ribosomes to be translated into proteins. Human host cells are eukaryotic. Eukaryotic cells commonly produce one messenger RNA strand per polypeptide. Within the start and stop codon on the mRNA strand is the code for one protein. This presents a problem for SARS-CoV-2, as its genome is one long positive sense strand of RNA that encodes for up to 29 proteins. 29 proteins must be produced from one strand of RNA containing one start codon. Thus, SARS-CoV-2, with an unusually large RNA genome of 27 to 32 kilobase pairs in length, must have specific mechanisms for producing such proteins from its single strand of RNA. For more information on the process by which the coronavirus produces its viral proteins, watch part 2 of this video.